okay? So 2, this is way simpler. All the rest are simpler than that one. Joining of small organic molecules into macromolecules. So now we have all those little monomers that are around. And in a 2009 study, they actually proved that those could spontaneously assemble into polymers, especially in the presence of hot sand, clay, or rocks. So when you have a hot substrate, something that these materials can sit on, it actually promotes assembly, which is kind of crazy, but scientists have proven that. They've gotten like hot sand, clay, or rocks, which all would have been available on early Earth's surface. Everything was very hot. And from these monomers that we just talked about, they can actually spontaneously assemble into polymers and form bonds, especially if the environment was reducing where electrons were kind of free and easy to pick up. And they will naturally be drawn towards one another because they have the same sort of chemical composition. So it's not a far stretch to realize that these would come together. So once they come together, now we have macromolecules. Now we're really cooking with fire, and we just need to start assembling these into cells in order to get some life to happen. And that is what happens next. So the assembly of macromolecules in something called a protocell. Okay, so what I want you to picture in your head is taking like a drop of oil and putting it onto water. And I'm sure you can visualize that in your head right now, right? If you have a drop of oil and you put it into a container of water, it's just going to form like a little bubble spontaneously, and it's not going to mix with water. Water and oil don't mix. You can shake it all up, right? And it will still form those like little individual oil bubbles throughout water, but it's never going to completely mix because water and oil hate each other. Oil is incredibly, obviously it's a lipid that's incredibly hydrophobic, but water is water, and so those two things don't mix. So if you can picture little droplets of lipids, which would have been present in early Earth, would spontaneously kind of form into little bubbles, just because if they're in water, they don't like that, they're very hydrophobic. Okay, so basically what happens is these little bubbles would trap whatever was around them in their environment. So if they're sitting in some sort of like soup of amino acids and nucleotides that have been formed into kind of maybe some macromolecules, and lipids get dropped in and then just kind of spontaneously form their little bubble, they're going to take a little part of that soup and those organic materials into that bubble with them. And this is a really, really important step to forming life because now we have all of our materials in one close location. Like if your parents make you like sit down at the kitchen table and force you to talk to your sibling to come up with a solution, now all of these things are forced to be in a really tight area. And now we can start seeing things like a metabolism or replication occur because they're not like all just in this giant pond somewhere on the surface of the earth, but they're in a nice enclosed space. So this diagram is important because it just shows the type of clay that we believe to be present on early Earth. They call Montmorillonite. Your guess is as good as mine. But this clay actually helps, and you can see this, the relative increase in the production of precursor molecules, and then therefore protocells in this time. So it's kind of going back to what I said before, where like hot sand, hot clay, and hot rocks serve as kind of a catalyst to speed up those reactions. Okay, so these little lipids, just like we have the lipid bilayer, and we'll talk about that a lot shortly here, I think next week. We have this lipid bilayer that's wrapped around our cells. Lipids were the same outer protection of protocells. Proto, think of like prototype, like the original kind of precursor to the real deal. And that's what's happening here. And now we have the means to actually have all these materials in one small place, therefore can bump into one another, and we can see the magic start to happen. Protocells, that's another picture for you of those little bubbles and how they would form. Okay, last step, creation of self-replicating means of inheritance. Wow. Okay, so this is what you need to take away from this. You need to know that scientists are 99% sure that while they used to think that DNA was the first method of inheritance because it just made more sense, if you really think about it, it makes a lot more sense that RNA would be the first actual piece of inheritance, genetic material. So the RNA World Hypothesis is pretty widely accepted by scientists everywhere. And there's a lot of different reasons for how scientists think, or why scientists think that RNA instead of DNA was the inherent material, the first genetic material. Wow, words. But But one, and most importantly, is that scientists have proved that RNA can serve as its own enzyme, which is crazy. DNA cannot, but RNA can serve as its own enzyme. They actually call it a ribozyme, and it can make short replications of RNA. So I can't even go into the details of how that actually occurs, because that's like super high level, like doctoral and past level of biochemistry. But scientists have been able to prove in the lab that RNA can actually kind of serve as its own enzyme and replicate itself. Okay. What you need to think about then to make even more sense of this, RNA is single-stranded and relatively stable on its own if it has like certain base pair combinations. Okay. So it makes a lot more sense that we would start with a single-stranded, simpler genetic material that's actually capable of its own reproduction or own replication in certain circumstances, than to think about DNA, and we will talk about this for sure when we get into talking about DNA, which has to be unzipped, which then has to have another enzyme that comes and replicates it. There's a lot of different things that have to happen for DNA to replicate. So the fact that RNA is really simple and single-stranded and can sort of spur that own reaction and by itself makes it a way better choice for probably what the first genetic material was. Okay, so now we have genetic material, we have macromolecules, we have all of these things inside of our protocells. They are going to, over time, spontaneously create reactions that are going to allow them to replicate. And then after a very long period of time, we will actually see life beginning. Right? But these are the steps, the precursor steps that have been taken in order to make basically the first cell. So if you can picture like a cardboard drawing of the first iPhone, that's basically what we just created. And over time it develops and it gets more and more advanced. And then we see life on Earth. So here we go. Love this analogy. So I'm going to reference this a bunch tonight because it's one of my favorite analogies because it just provides a really strong visual. But we can actually picture a football field, so all 100 yards, okay, as the timeline of the Earth, and it allows us to actually kind of visualize in our mind how long these things are taking to happen and how far away we are now from the start of the Earth, okay? So at one end zone, we would put the origin of the Earth. So this would be 4.5 billion years ago. So our Earth formed 4.5 billion years ago at the earliest. We have some data that says it could actually be a little bit older, okay? And then up here at the 15-yard line, which is going to be like 3.8 billion years ago, is when we have life forming. Okay, so it took a whole 15 yards, right? It took a whole like almost 1 billion years from when the Earth first formed for us to actually get life to happen. And we're gonna revisit this a lot as we add on other things and see more complex life occurring. But yeah, so keep this visual in your mind. But it took, yeah, a whole 15 yards just to get life to begin from the origin of. All right, how do we know all of this stuff? And how do we know like 4.5 billion years, 3.8 billion years? Great question. It's hard to know for sure, right? And again, we weren't there, so. But the way that we can do this is by measuring isotopes, okay? What an isotope is it's basically a version of a compound that just has a different number of electrons. So we have like carbon 12, electrons and neutrons. We have carbon 12 and then we have carbon 14, which has 2 extra components to it that makes it a little bit bigger. And these things just kind of occur naturally. But if you have something that has an isotope that has some extra either protons or neutrons or electrons in it, it will break down over time. And we know how long it takes those substances to break down because we have labs and we've watched it happen in real time. So what we can do is we can actually extrapolate or work backwards, right? So if we know that it takes 40 years for carbon-14 to half in size, we can take a look at like old school fossils or old school rocks and determine how much of a substance they have left in them, and then kind of work backwards to see how old that substance is based on how long it would take for that thing to break down based on its half-life. So I know that was like a really brief explanation there because it's not super, super critical. But if you're like, what in the world, how are scientists figuring this out? That's how. It's by looking at how basically substances or compounds have broken down over time and using rules that we know to be true based on what we've seen in the present day. And then kind of working backwards from that and seeing like, oh, if this thing has 1 gram of this left, then it probably started, 4 billion years ago with 100 grams or something like that. So that's how we're dating all of this stuff, or a lot of this stuff. And then there's other methods as well that scientists have developed over the years that are incredibly precise. But this is definitely one main way that this happens. Okay, so we're going to jump in now. So we've talked about how we got life on Earth. We've talked about what that looks like on our football field. And now we're going to start adding some more things to the picture. So this is like a clock, basically, but it puts the origin of Earth right here at about 4.5, 4.6 billion years ago. Okay, and then as we move around the clock, it takes like about a billion years, not quite, for prokaryotes, or those single-celled organisms to appear. Okay, and then you'll see it takes another really long time, basically a billion years, for oxygen to appear. Okay, and then it takes even longer for single-celled eukaryotes to appear. Okay, so prokaryotes versus eukaryotes, I have a picture up there, but prokaryotes are going to be like our bacterial cells that we think of today. They don't have any membrane-bound organelles. They don't have a nucleus, none of that. So it actually took a really long time for eukaryotes to come around, and then multicellular eukaryotes after that. So this is a really helpful graphic. We're going to be talking through these individual points over the next couple of slides, but definitely come back to it, and you can see kind of the progression and just how long everything took to get to where we are right now. Okay. All right. So next in line. So again, if we look back here, we just talked about how prokaryotes were. We talked about the origin of Earth. We talked about the appearance of prokaryotes. Now we need to talk about oxygen, because obviously we breathe oxygen, and most everything on Earth needs oxygen, except for some of our anaerobic friends, like the bacteria in your gut and things like that. But it didn't come around for a really long time, like a really long time. Like almost 2 billion years is how long it took for oxygen to appear. And I'm going to show you on that football field example, but it's like almost half of Earth's existence was without atmospheric oxygen, and then oxygen appears, okay? Just totally nuts. And it accumulates to the point where we can actually form an atmosphere like we have today. Like the oxygen just isn't like floating off into the rest of the solar system. So this is called the oxygen revolution. Again, it happened about 2 billion years ago. And basically what happened is we had things called cyanobacteria. So some of my students, and it's kind of a common misconception, but don't let this happen. They'll be like, oh, well, plants created the oxygen on the planet. Okay, and then I would refer them back to this picture, because we're still living in just prokaryote time. We don't even have eukaryotes, let alone multicellular eukaryotes, let alone colonizing land and doing all that planty plant stuff. Okay, so actually the first photosynthesizers, right, which is how we got oxygen on our planet, are what we call cyanobacteria, C-Y-A-N-O bacteria. And those are photosynthetic bacteria, and we still have them on the planet today. But we basically just started to see this rapid increase, right? And basically what happened is, almost like natural selection, the things that could tolerate the oxygen survived and figured out how to use oxygen or create oxygen, and the things that couldn't died. So we actually saw like 75% or more of species that were alive, or not species, but cells that were alive prior to the oxygen revolution died because they couldn't hang. Oxygen to them was very toxic, which sounds crazy because obviously we need it to survive, but it was very toxic. And so the only things that really remained were things that could either tolerate or create oxygen themselves, which is a really, really important time that comes next on our little journey. Okay, so now we actually have an atmosphere that has enough oxygen that things can utilize it. And now we have to wait like another, I don't know, 800 million years to get single-celled eukaryotes. Okay, so everything takes a really long time. We can't even fathom it. Okay,